Dear learners, today we will discuss about nucleic acids. Nucleic acids are long chain polymeric molecules. The monomer, that is the repeating unit, is known as the nucleotides, and hence sometimes nucleic acids are referred to as polynucleotides. Deoxyribonucleic acids and ribonucleic acids are two major types of nucleic acids. DNA and RNA are responsible for the inheritance and the transmission of specific characteristics from one generation to another generation. The nucleic acids function. Nucleic acids are responsible for the transmission of inherited characters from always the parents to offspring. They are responsible for the synthesis of protein in our body. That is, it's nothing but that is the central dogma. First, initially they are the productions of daughter DNA from parent DNA to daughter DNA to RNA, finally productions of protein. So final the productions of protein it needs for nucleic acids. Apart from this, responsible for the synthesis of protein, it also helpful for the DNA fingerprinting is a method used by forensic expert to determine paternity. It is also used for the identification of criminals. It also played a major role in studies regarding biological evaluation and genetics. The classification of nucleic acids, nucleotides, nucleosides, and phosphoric acid. First, the nucleic acid is nothing but that is it consists of polynucleotide. Polynucleotide is nothing but the combinations of nucleosides as well as the phosphoric acid. The nucleosides it's nothing but that is a nitrogenous bases and sugar molecule. The sugar molecule first that is they are the two part. One is the ribose sugar as well as the deoxyribose sugar. Nitrogenous bases purine and pyrimidine. The purine bases always the adenine, guanine. The pyrimidine bases cytosine, uracil, and thymine. Instead of uracil, they may be present in the thymine. It's called as a DNA. If they present in the uracil, means it's a RNA. Only one part is the difference. This structure show that the purine structures as well as the pyrimidine structure. Pyrimidine structures cytosine, uracil, and thymine. Purine, adenine, as well as the guanine. This structure show that the DNA as well as the RNA. RNA is nothing but ribonucleic acid. DNA, deoxyribonucleic acid. DNA or RNA, both of them they consist of the nucleotides. Especially for already you know that is a nucleotide is nothing but nucleosides as well as the sugar molecule. So nitrogenous bases, purine as well as pyrimidine bases, that is adenine, guanine, cytosine, thymine. It's called as a DNA. Adenine, guanine, cytosine, uracil means it's a RNA. So the DNA it is a double helical model of DNA. The RNA it is a single stranded of RNA molecules. Nucleosides. The nucleoside is structural subunits of nucleic acids, the heredity controlling components of all living cells consisting of a molecule of sugar linked to a nitrogen containing organic ring compound. In the most important nucleosides, the sugar is either the ribose or as well as the or deoxyribose. The based upon the presence of ribose as well as the deoxyribose sugar only they are conformed with the DNA or RNA. Cytosine, thymine, uracil, guanine. Cytosine, thymine, uracil. It's called as a pyrimidine nitrogenous bases, purine, adenine, as well as a guanine. This structure show that the nucleosides. Next one, nucleotide. Nucleosides as well as the sugar molecule. Like a nucleotide is nothing but it is an organic molecule with a basic composition of a nitrogenous base, pentose sugar, and as well as a phosphate. The DNA and RNA are polynucleotides which contain a chain of nucleotides monomer with different nitrogenous bases. Nucleotides are essential for carrying out metabolic and physiological activities. ATP, adenosine triphosphate. Based upon the phosphate group, they are called adenosine triphosphate. It acts as always the energy currency of cells. The nucleotides form various coenzymes as well as the cofactors like NAD, NADP, FAD, coenzyme A. The structure, a nucleotide, it consists of three units. One is a nitrogenous basis, that is a purine and pyrimidine basis. Second one is a pentose sugar, it's like a ribose as well as a deoxyribose. Phosphates, that is the based upon the number of the phosphate group will be present. It like maybe only one phosphate group, it's called as a monophosphate. That is having the A basis, poly A tail, and also the having the coding region. One is first the coding region also having the starting site as well as a stop codon site. Starting site U starting site AUG, stop codon site UAA, UAG, UGA. mRNA function, always the mRNA it involved to the genetic information from DNA to RNA and to protein like a central dogma. Because DNA cannot be decoded directly to produce amino acid, so it needs for RNA. mRNAs act as an intermediary between the DNA and protein molecule. Each piece of mRNA encodes the information for one protein in eukaryotes. However, the protein is the production from the amino acids. Every three nitrogen containing bases like AUG. AUG it's one of the codon. It will be helpful for the productions of one type of amino acid. All the amino acids join to form a protein molecule. So the main role, intermediary step between the DNA and protein. So mRNA is the main role for the productions of protein molecule. So initiation, elongation, termination, all the process it occurs in the translation process. rRNA, the RNA found in ribosome, the molecule responsible for catalyzing protein synthesis is known as a ribonucleic acid. Over 60 to 80% of the weight of the ribosome is composed of ribosomal RNA, essential for all of the ribosome's activities, including binding to mRNA, attracting tRNA, and catalyzing the formation of peptide bond between amino acids. These are the rRNA structure. They having the two type of the subunits. One is the large subunit and small subunit. Types of rRNA in prokaryotes. In prokaryotes, the 16S ribosomal RNA is housed in a compact 30S ribosomal subunit. The two rRNA species are present in the large 50S ribosomal subunit, 23S as well as 5S ribosomal rRNAs. So it consists of the rRNA types in the prokaryotes, 16S, 23S and 5S rRNA. In eukaryotes, it consists of the 60S as well as the 40S subunit. Usually have several varieties of the rRNA genes arranged in repetitive sequences, like the 60S subunit contains 28S rRNA, 5.8S rRNA, 5S rRNA, whereas 40S subunit contains 18S rRNA. The functions of rRNA, protein synthesis is the primary functions of rRNA. By attaching to messenger RNA and transfer rRNA, this molecule assure that the codon sequences of the mRNA is approximately translated into the amino acid sequences of protein. Finally, the difference between DNA and RNA. DNA sugar moiety in deoxyribose. The RNA, the sugar moiety is ribose. The base is present in the DNA, adenine, guanine, cytosine, thymine, RNA, adenine, guanine, cytosine, uracil. The DNA molecule always the double stranded molecules. RNA, it is a single stranded molecule. It is DNA, it will obey the Chargaff rule. Already you know that the Chargaff rule is nothing but adenine always pair with the thymine, guanine always pair with the cytosine. But the RNA is also they having the Chargaff rule, but it does not obey the Chargaff rule. Bases are not modified. Bases are modified in RNA. It is stable and not hydrolyzed easily by alkalis. It is unstable, hydrolyzed easily by alkalis. DNA content in constant in all the cells except during cell division. It varies from cell to cell. The lifetime of DNA is comparatively high. RNA is a very short lived. No natural DNA is catalytic. RNA can be catalytic. It present in the nucleus, mitochondria and chloroplast. It's a DNA. The RNA is always present in the mitochondria, nucleus, nucleolus, ribosome and cytosol. Thank you.